Hi, my name is Andres Ornelas, and this is Lola. She's here to give me brownie points. And today we will be talking about signal transduction. In the most basic way, signal transduction is how cells talk to each other. It is the method of communication between cells, obviously, and it is needed when a hormone or a neurotransmitter cannot pass through the cell membrane. Signal transduction starts with the chemical message and then it is transduced to an action within the cell. This happens by extracellular signals coming into the cell, and then they become intracellular signals which then tell the organelles how to respond. There are two types of signal pathways, one being the protein modification, and this starts off with the messenger, then goes to the receptor, which is then transduced and sends out the secondary messages, which are then received by the cell targets, and then that leads to the reaction. Then we have the phosphorylation cascade, which in turn becomes the protein kinesis, and that is the majority part of what signal transduction is.
Okay, so one of the membrane-bound receptors is the channel-linked receptor. So channel-linked receptors are cell membrane-bound receptors that are also called ligand-gated channels. So when a ligand binds to them, a sort of tunnel is opened in the membrane that allows a specific molecule in. The molecules that pass through them are ions. After a short amount of time, the channel is ready to bind to a new ligand.
G-protein coupled receptors are the largest and the most diverse group of membrane receptors in eukaryotes. These cell surface receptors act like an inbox for messages in the form of light energy, peptides, lipids, sugars, and proteins. Such messages inform cells about the presence or absence of life-sustaining light or nutrients in the environment, or they convey information sent by other cells. GPCRs interact with G-proteins in the plasma membrane. When an external signaling molecule binds to a GPCR, it causes a conformational change in the GPCR. The changes then triggers the interaction between GPCR and a nearby G-protein. G-proteins are specialized proteins with the ability to bind the nucleotide, guanylate triphosphate, GTP, and the GDP. Some G-proteins, such as signaling protein RAS, are small proteins with a single subunit. However, the G-proteins that associate with GPCRs are heterotrymic, meaning that they have three different subunits, an alpha, a beta, and a gamma.
Now, enzymatic linked receptors. These receptors are linked to enzymes located in the cytoplasm. They contain more than one subunit. When activated, the subunits fuse together. This is called conformational change. These receptors have effects which last for minutes to hours. An example of an enzyme-linked receptor is insulin. When insulin binds with two of its subunits, they fuse together, which is called conformational change. The activated receptor phosphorylates. Tyrosine resides on itself and other proteins.
Yes, hello, we are back, and now I'm going to be talking about soluble receptors with Lola. Lola, say hi. Say hi. She's not in the mood. Okay, so soluble receptors. An example would be intracellular receptors, and these are small lipid soluble molecules. They can readily cross the cell membrane. They interact with the receptor protein. So in a sense, these are receptors, meaning they're like, let's say, the enzyme, which will catch the protein, and now that they are together, they can pass through the cell membrane. A type would be an aldosterone, and it is a lipid-soluble hormone, and this can easily diffuse through the plasma membrane, and once inside the nucleus, it binds to the DNA. And that is a soluble receptor.